about epitope mapping, basically where we're looking at a protein alone and a protein with its antibody. Now, this application is slightly different from a typical protein-protein interaction because here we're not always interested in getting the peptides back from the antibody. If you do a protein-protein interaction, you often want to get peptides from both proteins and map the interface and then potentially model it. But here we're not necessarily that interested in the antibody, so it makes it a little bit easier. But then also antibodies have these disulfide bonds, and so you have this added step of making sure you really reduce those disulfide bonds before you put your sample in the mass spectrometer. But again, many have come before us, and there are many protocols and papers available to guide you through doing this. So this is work that we did with the Maynard Lab at UT Austin. And I want to point out this was done very, very quickly by a first-year student in my lab. The reason I want to point that out is because often professors are worried that it's going to take students a long time to learn how to do an HDX experiment. This student had no prior experience with HDX or mass spectrometry, and I think in the space of about 4 to 5 months was able to produce this really, really nice data. I will say there was a bit of pressure because we were epitope mapping the coronavirus spike protein. So pathogenic coronaviruses are something which I'm sure all of us have heard of because we're in the middle of a SARS-2 coronavirus outbreak. But there are other coronavirus, and there have been other outbreaks as well. For example, there was an outbreak of SARS-1 in 2002 and MERS in 2012. So the goal here was to see if we could identify pan-coronavirus epitopes that would act on all of the different coronaviruses that are pathogenic to humans. Now, in order to do this, we targeted the spike protein because all of these coronaviruses use the spike protein to mediate fusion with their target cell. So the spike protein is shown here in panel A. It is a trimer, a homotrimer. It has an S1 and an S2 domain, and the S1 domain is cleaved from the S2 domain following the S1's domain interaction with the ACE2 receptor. Now, once this cleavage occurs, the S2 domain then undergoes a conformational change and then mediates fusion with the target cell. Now, all of the coronaviruses have this spike protein. And so our question was to identify epitopes specifically in the S2 domain rather than in the S1 domain. So most of the neutralizing antibodies that you're probably reading about in the media and in the literature, they really focus on this S1 domain and specifically this this receptor binding domain that mediates this interaction with the ACE2. But we were focusing on the S2 domain because it is more conserved between the various coronaviruses. So our collaborators had immunized mice with a stabilized MERS2 S2 protein. Okay, so this was not even the coronavirus, it was not even the spike protein from the SARS-1, which causes the coronavirus. The resulting antibodies were then characterized, and 3 of them were identified to bind all 3 spike proteins. So the spike protein from SARS-1, SARS-2, and MERS. Of these 3 antibodies, 1 antibody then showed neutralizing activity in cell fusion and pseudovirus infection assays. So in vitro assays, this antibody showed a lot of promise. We also showed that the antibody was conformationally sensitive. In other words, if we denatured the spike protein, it would no longer recognize it. But then we got stuck because even with one of the world's experts on cryo-EM of the spike protein, we were unable to get a structure of the 3A3 antibody bound to the spike protein. So the question was, can we identify the epitopes specifically of 3A3 using hydrogen deuterium exchange, and could we do it very, very rapidly? So this is the coverage map that we obtained for the spike protein. The goal was to do the spike protein alone and then compare it to the spike protein with the antibody. Now, for anybody that already has HDX experience, you might look at this and think this is a horrible coverage map. The reason I say that is you can see we have large regions of the spike protein for which we don't obtain any peptides. Now, the reason for this is that the spike protein is very heavily glycosylated. The glycosylation sites are shaded in gray here. And we did not allow glycosylation as a modification when we searched for the peptides. Now, the reason that we did this is because we had some data that showed that the 3A3 antibody had the same affinity if the spike protein was glycosylated or not. And we wanted to work quickly, right? We wanted to really focus down on as few peptides as possible to figure out what the epitope was. So we went forward. Now, these are the data from the non-neutralizing antibodies, the 4A5 and the 4H2, and we were unable to identify their epitopes. So this tells us that their epitopes are probably in a region where there is a glycosylation, but of course we were not so interested in identifying their epitopes as they were not neutralizing. But just to take a step back, these are those volcano plots that I mentioned. So on our x-axis, we have our delta change in HDX. So the spike protein alone, uptake minus the spike protein with the antibody. And then on our y-axis here, we have our p-value. So for all our experiments, we do 3 or 4 replicates, and that enables us to determine a 95% confidence interval. So if there was something that had a significant change, we would be seeing dots here. And here we have the uptake plots. And again, you can see that in all conditions, the amount of deuterium uptake is the same. So we were unable to identify their epitopes. But of course, I wouldn't be telling the story if that was the case for the 3A3. So here are the data for the 3A3 antibody. We actually had an IgG as well as a Fab. And so in panel A, we're showing the data for the IgG, and in panel B, we're showing the data for the Fab. And you can see it, I just chose one time point to show here the 100-second time point, that we're getting a collection of overlapping peptides all in this significant region. And then if we look at the uptake plots, you can clearly see a difference between the black, which is the spike protein alone, and the orange and blue, which is with the IgG and the Fab. So now if we map this onto the spike protein structure, so this is the structure of the monomer. In blue, I'm showing the data for IgG. In orange, I'm showing the data for the Fab. They're essentially identical, but you can see we clearly localize the binding or the epitope to the apex of this S2 domain. If we look at the sequence of this epitope between 7 different coronaviruses, we can see that it's highly conserved. And if we look at the structures of the spike proteins from these coronaviruses, we can also see that it's structurally very conserved. So we were able to very rapidly identify the epitope of this 3A3 IgG. Now, what's interesting is if we look at this HDX data in the context of the spike protein in its trimeric form, here, again, I'm projecting the HDX data, but in gray, I'm showing regions of the protein for which we had no coverage, right? Remember, that was quite a lot because we didn't allow for glycosylations. We can see that in the trimer, when it's in this closed state, so that's when the receptor binding domains are all in the down state, we cannot have access to our epitope. And as we progressively move these receptor binding domains to their up position, these are different cryo-EM structures that are available. Now we can see our epitope is becoming more available. Before finally in this last structure here, we actually have the ectodomain of the ACE2. We can almost completely see our epitope here. So this tells us that this 3A3 epitope is conformationally sensitive, and perhaps explains why we were not having much luck with the cryo-EM. So if we put this in the context of this conformational change of this S2 domain, we start with the S1 and S2 as a single protein. It's the S1 that's going to interact with the AC2 receptor, leading to the cleavage of S1 from S2. Here, that would lead to an exposure of our epitope in this prefusion, as it's described, conformation, which would otherwise subsequently undergo this conformational change. This would be our epitope here, which would now no longer be recognized by our antibody. So we really have identified, a potential epitope here that can be used for a pan-coronavirus therapy. So the S2 apex may be a vulnerable site for pan-coronavirus neutralizing antibodies, but I guess more generally, HDX can be used to rapidly screen for antibody epitopes. Again,